Hello everyone. In today's class, we'll study about the sexual reproduction in flowering plants. So flowering plants means angiosperms, right? Angiosperm. This is the most advanced category of plant kingdom. Now what is a flower? Flower is a modified stem, or we can say modified shoot also, which will function as a reproductive organ and produces the egg or pollens. So how it is happening? To understand that, we need to study the structure of flower or the parts of a flower. Okay, this is the basic diagram of a flower, wherein here this part is called as pedicel. Okay, this part is called as a pedicel, which is nothing but the stalk of a flower. Stalk of a flower. Okay, if the stalk is present, then the type of flower is called as pedicellate. If the stalks are not present, then the type of flower is called as sessile. Right? This is the basic term where the stalk of a flower is called as a pedicel, and if the pedicels are present on a flower, then we are calling it as pedicellate, and if the pedicels are not present, then we are calling it as sessile. Okay, coming to the next part here, this part is called as thalamus. This part is called as thalamus, on which this is a stage on which the flower is placed. The total flower is placed on this thalamus. Right. Now we'll come to the 4 different whorls of a flower here. First is the calyx. Okay. The first whorl of a flower is nothing but the calyx, which we can say it is the, as I told, it is the first whorl, and this is green in color. Green in the sense, if it is green in color, then definitely it will perform the process of photosynthesis, and also it is the outermost layer, because of which helps in the protection. What is the function here? Which is helping, which is having a protective function. So calyx is the first whorl in the flower, and it is green in color, because of which it performs the process of photosynthesis, and it is the outermost layer, and hence it is protective in function. Right? Next, move on to the next whorl, that is corolla. Here, right, this is the corolla. This is the second whorl, and this attracts the pollinators, right? Because it has having a bright color, and also because of the good smell, this corolla will attract the pollinators. Okay, now this is the second whorl. And when we are talking about the third whorl, that is, before going to that, let me explain here the parts. This is here, this part is called as the anther. Here you can see here. Then the down part of that one is called as filament. The down part is called as filament, and together we are calling it as stamen, which is nothing but the unit of androecium. Stamen is the unit of androecium. Androecium is the male reproductive structure in the flower. So what is the unit of androecium? Stamen. This androecium is the third whorl and will represent the male reproductive structure in plant. Right. Now next moving on to the other part, that is the female reproductive part. Now where it is, this part is called as stigma. Now this is a part which will receive the pollen. That means it's a landing for the pollen. It's a stage where the pollen will land here. And this is the style present just beneath the stigma. Okay, this is the style just beneath the stigma, which is a long slender tube-like structure from which the pollen will travel to reach the ovary. Now you can see here the ovary. Here the down basal part, we are calling it as ovary. Together these 3 parts, we are calling it as carpel. Okay, and the carpels are nothing but the units of gynoecium. Units of gynoecium, which is the fourth whorl. Which is the fourth whorl. Now we finished the first whorl is calyx, second is corolla, third is androecium, and the fourth one is gynoecium. This is the fourth whorl, which represents the female reproductive part of a flower. Right, these are the four whorls of a flower. Now how we are going to denote this? Calyx is represented by K, corolla is represented by C, androecium is represented by A, and gynoecium is represented by G. These are the symbols which we are using to represent the four different whorls. Now I have shown that the units of androecium are stamens and the units of gynoecium are carpels. The units of corolla are petal. What are the units of corolla? They are petals. In the same way, the units of calyx are nothing but the sepals. Right? Now these two, calyx and corolla, they are called as non-essential whorls. What we are calling? Calyx and corolla. These two, we are calling it as non-essential whorls because they are not participating or they are not directly involved in the sexual reproduction. They are also called as accessory or non-essential whorls. Okay, then when these two are non-essential whorls, then what will be the essential whorls here? The essential whorls are nothing but the androecium. Okay, I'll write here androecium, and in short I am writing here androecium and gynoecium. These two are called as essential whorls because they are directly involved in the sexual reproduction. So now we'll discuss about the stamen. The stamen is the unit of androecium. Unit of androecium, which will represent the male reproductive part of a flower. Right? And this is otherwise called as microsporophyll, which is otherwise called as microsporophyll. Phyll means leaf. That means a leaf which produces the microspore. Microspore is used for particularly for the male reproductive organ or male reproductive part of a flower, which is nothing but the stamen. Now each stamen, this is the structure of a stamen. Each stamen consists of two parts. The first part is called as anther and the second one is called as filament. The anther and the filament. This is the distal end. This is the distal end, and here this point at the end where the filament or where the total stamen is attached with the thalamus, we are calling it as proximal end. Proximal end. That means this proximal end of the filament will attach to the thalamus of the flower, whereas at the distal end you can find the anther. Together these two parts are called as stamen. Now the anther is a bilobed structure. Bilobed means two-lobed. It is a bilobed. Bi means two. It is having two lobes. Now in each lobe you will find two theca. Two theca means two sacs. Each lobe will have two theca, which is called as sac. Sac means it's a bag-like structure, and hence the anther is called as dithecous in nature. Yeah, you can see here this is the first lobe, right? This total together will form the first lobe and this will form the second lobe. Now these are nothing but the theca, right? The anther has in each lobe it has two theca, that is sac-like structures called, and hence it is called as dithecous. Now there are certain species of plants where the anther will be only having single lobe. Okay, that condition we will call it as monothecous. Monothecous in certain species. Like example, you can find this condition in case of hibiscus. Okay, now this was about the dithecous and monothecous. Now if you see the 3D cut section of an anther, you can find here 1, 2, 3, 4. 4 cavities. Okay, these cavities are also called as microsporangium. That means in each lobe, 2 cavities you can find here. 1, 2, 3, 4. So total 4 microsporangiums you can find, and hence the anther is said to be a tetrad structure, or we can say the anther is a 4-sided tetragonal structure located here at the corners. Now see here, 2 at the corners here, and then again these 2 also at the corners, and also 2 in each lobe. एक lobe में 2. So 2 in each lobe. Now you can see I have drawn a wall or a sac-like structure here connecting the 2 lobes, which we are calling it as pollen sac. Pollen sac. And inside this pollen sac we can find the pollen grains. That means these 2 cavities or the microsporangium will consist of pollen grains, right? Now these are nothing but these here 3D section. These are the lines of dehiscence. Dehiscence means the rupture because of which the release of pollen will occur. These lines of dehiscence. Here also if you see, there is one line of dehiscence here, right? And these lines will have special cells called as stomium cells. Stomium cells, which are present only at the line of these anthers or these lobes. Now these stomium cells which help in the process of dehiscence. What is dehiscence? Dehiscence is nothing but the release of pollen. Now let us see the transfer section of a young anther. If you see this structure, there are different layers. The outermost layer we call it as epidermis, okay, which will protect and help in the dehiscence of anther to release the pollen, right? And the innermost layer, the second layer is called as endothecium, which will lose the water and contracts at maturity, and then hence that is helping in the dehiscence of pollen again here. Then the next layers are here, the middle layers. We didn't say middle layer but middle layers because they are 1 to 5 in number. The middle layers are 1 to 5 in number. All these 3 layers, epidermis, endothecium and middle layers, they will protect and also help in the dehiscence of anther. That means in rupturing of pollen to release the pollen grains, right? The next layer we have the tapetum. It will help in nourishing the developing pollen grains, right? Now these tapetum, here you see the enlarged view of this one of the microsporangium. Only if one part, if you see, we are enlarging it here, and you can see the epidermis, which is only one single layered, right? Endothecium is a compactly arranged radial cells you can find here. Middle layers 1, 2, 3. Now here you have find 3 middle layers. In some species you can find more than 3. 1 to 5 layers can be present. All these 3 layers, epidermis, endodermis and middle layers, as I told you, they will help in the dehiscence of anther to release the pollen. Now apart from these, tapetum, this will help in nourish the developing pollen grains, and this tapetum is contains or these cells possess the dense cytoplasm. Here you can see the cytoplasm is very dense and it will have more than one nucleus in the cells. Now this tapetum also contains a special organic material called as sporopollenin. Sporopollenin, which is a very hard structure and also can withstand the high temperatures. And from here, one more substance is present called as callase. Now this callase will release an enzyme called as callase here. Callase spelling, then it is called as an enzyme. This enzyme will help in degrading or which in breaking the sporopollenin and helps in dehiscence of anthers or in rupturing of anthers. Now apart from this epidermis, endothecium, middle layers and tapetum, we also have sporogenous tissue, right? This tissue is present inside the cavity, that is nothing but the microsporangium, okay? These are a group of compactly arranged homogeneous cells, that too when the anther is at the young stage, right? This stage, these tissues are called as sporogenous tissues and which occupies the center of each microsporangium. This is present in the center of the microsporangium only and when the anther is young. Now this is the figure which is showing a mature dehiscized anther. That means all the pollen grains are released because of the rupture in the cells. And here are the stomium cells. Stomium cells are nothing but the cells which are present at the lining of the anther. This is the lining. Here pink color, you can find these cells, the stomium cells. In the next class, we'll continue with the microsporogenesis. Thank you.